And so with that, I would like to invite our very first speaker to the podium. Is that right, Mary Lee? We're good. Okay. I'm looking on the screen. There's nothing. I'd just like to say a few words about Dr. Thea Tlsty, who's a professor at UCSF. And I will tell you that the words genomic plasticity, the very first time I heard those words were from her lips. For decades, she has been studying genomic plasticity, and she was one of the first to appreciate that the work that she was doing was, in fact, being driven by plasticity of the genome. Obviously, over time, this has now been emerging as a common phrase in the lexicon, but the perspective that she brings to this field, I think, is unique because of the depth of her understanding and the fact that she has really been one of the pioneers in the field. So with that, let me invite Thea to the podium.
Good morning. It's really an honor and a lot of fun to be here. Interacting with this group was a blast, and I can't think of a topic that excites me more than this particular one. Cheryl was correct. I've been studying this subject for nigh on 30 years, and I've published in the area of telomeres, copy number variation, and transposons, and what I've been very interested in is how it all comes together. And so I was asked to give a conceptual talk, and that's what I'm going to do. It's backed by a lot of data, but I'm not going to go into the details. They're published. But what I wanted to do is to pull it together for you. So we know that people have been studying this subject for a very long time. We know that for decades now that you can have certain types of DNA damage and it breaks the chromosomes, or it makes pyrimidine dimers, or this adduct or that adduct. I actually studied that as a graduate student. We know that radiation can give you types of breaks. We're going to hear about replicative stress and how it can cause alterations in the genome. These are endpoints, and people study the endpoints that has been described as genomic instability or genomic plasticity. They've gone beyond that, and they've looked at specific changes and how they relate to diseases. And so I just threw a few examples up here. For instance, if one has a mutation in BRCA1, it gives you a susceptibility to breast cancer, mutation in hemoglobin, thalassemias, etc. And I put some examples up here. We're probably going to hear a lot more about it in terms of telomere alterations and some of those endpoints, retrotransposons, copy number variation. So many aspects, these aspects of genomic instability or genomic plasticity are well studied. And the reason that we study them, some of the reasons that we study them, is to try and get a handle on evolution, disease risk, phenotypic variability, and ethnic susceptibilities. And when people study these subjects, what they do is they look for interpersonal variation, how different groups of people vary in terms of these endpoints. And as Cheryl said, we once thought that the genome was static, but now we appreciate that it's fluid. And I really chose this picture very carefully, as you'll see during my talk, because I think it really gives a nice illustration of what we're going to be talking about. And so we're starting to understand the extent of the transitions between states. So this isn't endpoint 1 and endpoint 2. It's actually very fluid states that go back and forth. And that's why I thought an iceberg was particularly beautiful to illustrate this point, because you have water molecules that adhere to the iceberg and can freeze and add to it, and those that can come away. And one molecule can be in either state, back and forth, back and forth. It's not relegated to one state. So we can study now the action of genomic plasticity rather than the end point. We can study the verb rather than the noun. And that's what I'm going to try and talk to you about today. So I'm going to talk to you about a new subject for my laboratory and some work that's just being published right now. We have about 3 or 4 papers that are just coming out starting to look at the underlying dynamic stochastic events that generate genomic plasticity. And what you start to realize is this is an ongoing process, and that there's a lot of intra-personal heterogeneity that's being generated. This subject is very much tied into environmental stress. There's whole programs that are turned on in response to stress. And I would be remiss if I didn't mention something about contingency loci. And these are areas within the genome that are hyper-variable, hyper-mutable. And they actually flip at different frequencies just for the fun of it. Now, you can increase that flipping by exposure to environmental stresses. But even in the absence of that, just every once in a while, they flip. And I'll talk about the ramifications of that, although I do have some of them up here. So, for instance, cell surface receptors to help evade the immune system. You can have class 1, class 2, class 3, if you want to call them that. And so when the immune system starts to mount a response to class 1, a certain part of the population simply flips to class 2 or 3, and they're now protected until the immune system catches up and then targets that, and they flip to a different one. So this has been known for quite a while, and it's a very effective way of surviving an immune attack, prey, predator, etc. Pluripotency genes, pluripotency proteins. It turns out that the stochastic variation is widely used in terms of trying to understand cell fates. I'll give an example of that. Okay. So as I mentioned before, what I'm going to talk about is intrapersonal variation and not so much interpersonal variation. I want to talk about the changes within one person's body, and I want to emphasize that actually we're all mosaics. And the closer you look, the better our technology becomes for looking at single cells, the more you realize that we really aren't a homogeneous set of cells, even within one tissue, even within one cell type. There's many different changes, and it's also on the level of the genome, and which is quite surprising. So today I would like to discuss the underlying rate of stochastic variation and how these rates can actually generate genomic plasticity and how it responds to environmental stresses. And then what I want to do is to give you some examples of how this genomic plasticity can actually affect its surroundings. And not only affect its surroundings in one ripple of activity, but it can actually fold back and create more genetic plasticity. So you actually have a feed-forward loop that keeps going and actually cascades. And that has some consequences in terms of disease. And then I'd like to finish with a short story in terms of clinical utility, how by listening in on this genomic plasticity, we can actually predict what a tissue is going to do before it gets done. So, stochastic variation. So, what I'm showing you here is a DNA molecule, and these are transcription cofactors. And what we've learned is that as transcription occurs, you have the proper coming together of the factors, and you have a productive production of RNA over there. And this is meant to mean that sometimes the cell has productive interactions. Sometimes it can go for a long period of time, as indicated by this. Here you have the same molecules, but they miss each other because of stochasticity. And so there's periods when a cell has a relative dearth of proteins, and there are periods when cells have an abundance of these proteins. And this is just one cell. So it goes up and down, which is what I meant by this wiggly line here for gene expression. So this stochastic variation can change protein numbers, and it occurs by chance, and it leads to phenotypic interconversions. So if you have stochasticity, you have timing of synthesis and degradation, and so you can have a cell that has 1x amount or 2x amount of a protein. Due to chance, it can drop to half, it can drop lower. Due to chance, it can double, it can even go up higher. So what it does is it actually forms a distribution, and accompanying that distribution of proteins are phenotypes. Okay? So the consequences of fluctuations can result in the generation of variation in observable traits. So I'm going to give you some examples, like, for instance, drug sensitivity, growth, cell fate determinations, etc. Let me give you an example. So here, what I've done is I've taken one cell, and it's doubled again, so now we have 4 cells. And the amount of protein that this cell has is shown in this very tight peak. But now, given the stochasticity that I was just talking about, some of these cells are going to throw the dice, and they will not produce as much protein as this tight peak, and some will produce more. And so what happens is you generate heterogeneity. It's phenotypic heterogeneity, and these cells can go from one state to the next in either direction. They can actually slide. A cell does not occupy one state permanently. It's transient, and it can slide amongst these states. Let me show you the ramifications of that. So, for instance, one of the things that I'm going to talk about a little bit more extensively is dihydrofolate reductase. It's a metabolic enzyme, and it's a target for chemotherapy. And so oftentimes, patients are exposed to the chemotherapeutic drug methotrexate, which binds to DHFR, dihydrofolate reductase. And if your body, for example, or your cell, has this amount of DHFR, it is resistant to X amount of methotrexate. If it has this amount, it's also resistant. But if the fluctuation has allowed it to dip a little bit lower so that now the drug is in excess of the protein, these cells, everything beyond this line to the left, will be sensitive to the drug. Those cells will not be able to use dihydrofolate reductase to make one-carbon transfers to synthesize DNA. Those cells will either arrest or die. And so by exploring this whole range of phenotypic space, the cell can generate cells that are resistant to a drug or sensitive to a drug. But what I'd like to emphasize is that this cell that was right now resistant to this drug, in a couple of hours or a couple of days, can actually fluctuate in this direction and become sensitive. And likewise, this one can go in that direction. And so this is becoming very clear in the literature right now that you can have a distribution of cells like this as seen by flow cytometry. And as I'm going to show you, you choose a cell from this area, and what happens is that this whole spectrum of heterogeneity is regenerated from that population of cells. I'll show you that in one moment. So this is looking at drug resistance, but now let's look at a different protein. Same concept. So here you're shuttling between low and high. Low, here, highest, higher, high, low. And the cells can go back and forth between these different states. And let's say that this is a sugar receptor. That means that if the cell finds itself in an environment which has ample nutrients, all of these cells will compete equivalently. They'll all live, they'll all proliferate. But if this cell or population of cells finds itself when nutrients are limiting, these cells will be at a selective disadvantage, and it's only these cells that have a lot of receptors and can gather the sugar that will be able to feed themselves and proliferate. So this is a way that cells can take the opportunity to explore phenotypic space. Cells use this mechanism for pluripotency and cell fate decisions. And here I wanted to give you an example of productive information on both ends of the spectrum. And so this is SCA1 content in stem cells. And if the cells have a low amount of SCA1, they have a preference towards erythroid differentiation. If they have a high amount of SCA1, what they have is a preference for differentiation in the myeloid direction. And so you can see that by exploring this space, the cells start to make decisions about which way they want to go in terms of expressing a phenotype. This is also seen with other pluripotency proteins, such as NANOG, etc. But now what I want to do is I want to tie this to genomic plasticity for you. So there's a set of recent papers that have come out recently by my colleagues at UCSF, but other people as well, indicating that you can have, if you have overexpression of replication proteins, these proteins are found at the origin of replication, what it does seem to do is to induce gene amplification. And these people have hypothesized that you get a re-replication event because these proteins are at the origin, and that there's subsequent gymnastics in the genome downstream. I'm not going to deal with the details of that. But what I do want to show you is that by fluctuating the amount of these replication proteins, this now gives you the opportunity to explore genotypic space, not only phenotypic space, but genotypic space. So here, if you have an amount of, if you have a low amount of one of these origin proteins, CDC6 is a really good one. It's very responsive, CDT1. If you have a modest amount of this protein, you go along and you have a very low frequency of generating copy number variants, duplications, or gene amplification. But if your cell has undergone one of these stochastic events and has temporarily, transiently, an increase in the amount of this protein, that is accompanied by a jump in copy number variation of several orders of magnitude. The cell only stays in that state for a short period of time, and then it comes back down here into the safe area. What this means is that any given point in time, this population is going to have some cells that every once in a while decide that they're going to amplify. And the amplified copies can then be cleaned from the system. What I'm trying to emphasize is that all of this is transient. The cell population as a community is trying out different solutions to whatever is happening in the environment at that point in time. So the increase in the amount of these proteins gives you a higher probability of duplications, amplifications, and copy number variants. The same thing happens with telomeres. It turns out that if you overproduce the telomere binding protein, TRF2, what happens is you don't have the proper formation of telomere ends. They become sticky, and now when they go through replication, you have ripping of the chromosomes, bridge breakage fusion, a la Barber-McClintock. And so with stochasticity, what you find is that every once in a while, at a certain probability, you have cells that have a high amount of TRF2, and these cells are at a higher probability of undergoing those processes and undergoing double-strand breaks. So this is how you can have phenotypic stochasticity. It can actually be tightly linked to genotypic stochasticity and genotypic plasticity. These fluctuations are ongoing. So is there a way that we can study them in real time? Most of the time when we study genomic plasticity, we take a snapshot. We grab the cells, we kill them, we look at the mutation, we look at maybe the phenotype that came out of them. But what would be really good to do in the case of what I'm describing to you is to be able to watch the cells as they're shuttling between high and low. Why do they stay in the high area for this amount of time? Why do they shift down? What kind of environmental factors can modulate? I'll talk about that in a couple of minutes. What kind of genetic factors can modulate this? In order to study these types of things, we have to use technology that's available right now, but is now being used for this purpose, single cell measurements. And this is actually coming along quite rapidly. But for a long time, we've had flow cytometry, time-lapse microscopy, etc. The things that I'm describing to you over the last couple of minutes cannot be seen when you look at the population as a bulk, because it all gets averaged together, and it all looks static unless you look at one cell and watch it moving from place to place. Okay? So, This is a technology that was actually developed in the early 80s, and one of our speakers, Randy Johnston, was important in developing this in the laboratory of Bob Shimke. And here I'm talking to you about dihydrofolate reductase, and as I indicated, dihydrofolate reductase takes folic acid to dihydrofolic acid to tetrahydrofolic acid, one carbon synthesis used for epigenetic modifications, used for DNA precursors. Very important molecule in the cell. And a long time ago, oncologists realized that if they could inhibit this drug, they could actually slow the growth of cancer cells. And so methotrexate was developed. And what it does is it binds to the active site of DHFR. So in the laboratory in the early 80s, what was developed is a fluorescent label for the methotrexate. It's compatible with cell viability. It doesn't exert a selection pressure because you can augment the media so that the cells don't see stress. And it allows you a single cell measurement of the DNA content in a cell. So for instance, you take a population of cells, you put in fluorescinated methotrexate, and the amount of protein that's within the cell is indicated by where the cell is in the flow cytometer spread. So cells that are out here bind much fluorescinated methotrexate. They have many proteins, and the ones down here are just the opposite. So this is a beautiful system for viably looking at cells that can undergo these fluctuations. So I want to show you one example of looking at this. This is a cell line. I'll talk about specific cell lines and primary tissue in a couple of minutes. But this is a generic cell line. And you can see that when you put in fluorescinated methotrexate, you see a spread. You see a coefficient of variance that's pretty broad. Now let's take one cell out of this population, or you can even take a small number of cells. And now what you have is a very tight coefficient of variance, right? It has a certain amount of protein. If you simply allow time to elapse as the cell duplicates and expands, what you find is that this spectrum is recapitulated. And it happens over a period of time. I'm going to show you that the period of time is cell type specific and can be modified by exposure to environmental agents. So here what I'm showing you is a high coefficient of variance. When you choose any portion of this, the coefficient of variance drops dramatically, and then over time it goes back up again. Now let's take a cut of these cells, and instead of taking it from the center, let's take it from the side. It could be from the left side or the right side. I'm going to show you this example. But what you're going to see is that this population reacquires homeostasis. So here, we're taking the whole population, and now what we're doing, as pioneered by Randy about 30 years ago, is we're cutting off the top 5% of the population, putting it through a flow cytometer, and now simply letting it grow for a period of time. And what you can see is that there's a shift to the left. First, the mean of the population is close to this cut point, but then you see that it shifts and it reinstitutes the original distribution. And here I'm showing this with an actual sample. Here, we're taking the top 5%. The mean has been shifted dramatically, but after a short period of time, the cells reinstitute this distribution. And so here you can see the mean is there. It becomes much higher when we do the cut, and then it goes back to its homeostatic set point. Each cell has different homeostatic set point, as I'm going to show in a moment. So these can be used to study the phenotypic variation, the stochasticity in terms of protein numbers, which is what I was talking to you about and what I just showed you in terms of protein copy number. But it turns out that you can also use this technology for looking at real-time genomic plasticity as it's occurring. Because, as I mentioned before, this was developed to look at gene amplification as it was happening. So here is the paper by Randy Johnston rapid spontaneous DHFR amplification as shown by cell sorting. And this really was an elegant experiment and way before its time. And you can see this is a population of cells. This is its distribution of DHFR protein. And what Randy did in a serial set of experiments is he took the top 5%, grew it up, took the top 5% again, did it again and again and again. Here's the second sort, here's the fifth sort, here's the 10th sort. And the really interesting thing that he did is he looked at the DHFR copy number. And you can see that as the sorts occur, what you're doing is you're obtaining cells with higher and higher copy numbers of dihydrofolate reductase. Going through this type of a procedure, he gets cells with 40 copies of dihydrofolate reductase. That's starting from a population of cells that does not have amplification. So this is a way that you can start to study these processes in real time. And this hasn't only been done for DHFR. Len Herzenberg's lab did it looking at a cell surface receptor. The very same trick, simply taking the top 5%, sorting it again and again, and obtained cells that were 50-fold amplified for this cell surface receptor. You can take the cells on the other side and sort in the opposite direction and get the cells to go back down that way. They will go in either direction. Okay, here's an example that I wanted to show you of a breast cancer cell line 231s. Here's the population with DHFR, fluorescent methotrexate, showing the distribution of DHFR protein in a population of growing cells. If you take the top 5% of the population and now go through a series of time points, and what I'm showing you now is a time point at 96 hours, you can easily see that this population has reinstituted the spectrum that it started with. That's in contrast to what I'm going to show you with this cell line, Chinese hamster ovary cell line. So here we do the same thing. We take the top 5% of the population, wait the same amount of time. I can tell you that these cells have the same doubling time and everything. And now what you'll see is during that same period of time, these cells have not shifted back to their original state. If you let them keep going for quite a while, I think it takes about 5 times longer until they finally reinstitute the equilibrium that they had previously. So there's differences in how different cells reinstitute their homeostasis. This has been done looking at different loci, and what you can find is that there's switching at different rates at different loci. You can take these cells and expose them to environmental stresses, and what it does is it accelerates the rate. This is dose dependent, it's time dependent, and what I'm going to show you is it's still cell type specific as well. And so here are the MDA 231s that I showed you previously, and I showed you that at a certain point in time they pretty much reinstitute the mean that they started with. But now what I'm going to show you is a primary set of cells. This is a population, not a cell line. It's taken directly from the human body. And when these human mammary epithelial cells are sorted for that 5% and then allowed to undergo the same type of conditions, you can see that it has very little shift back to the mean here. And this MCF10A, which is an immortalized but non-tumorigenic human breast cell line, you can see has an intermediate rate of shifting back to the mean. And what we've been doing is asking about how this relates to malignancy and the ability to transform, and we've populated this with multiple cell lines at this point in time, and what we're finding out is that switching rates seem to go up with tumorigenicity. Knowing this, we can now start to ask about genetic or environmental factors which can either increase or decrease switching rates, and we found genes that modulate these processes. not every spectrum of protein expression is a nice Poisson distribution. And here I'm just showing you a distribution of nanog protein in some stem cells. And it turns out that only when cells are in a certain part of this spectrum are they actually pluripotent. Otherwise, they're not. And so, given these biphasic peaks, how does the population, how is it able to actually keep the cell, keep this type of a profile? If you take a cell from here, let it go for a period of time, what you'll do is it will repopulate the double peaks. If you take a cell from here, let it go for a period of time, it will repopulate the double peaks. How is the population doing that? How is the cell making the decision to run up and down this continuum so that it has a distribution where this is low and this is much higher? Part of the answer to that has to do with the decisions that a cell makes. And so any one cell has several decisions. This cell, which I used the DHFR example, so I'm indicating that it's a sensitive cell. It has a low amount of DHFR. It has a mortality rate that's indicated by this factor. It has a replication rate that's indicated by this factor. And it has a switch rate that's indicated by this arrow. And so this cell has three choices. It can proliferate, it can die, or it can switch. Likewise with this resistant cell. It can die, it can proliferate, or it can switch. Those are its choices. And so the variables that impinge on these kinetics, on these different factors, are what helps to determine what the relative density of cells are in this peak versus that peak. There's another aspect of this that determines what this looks like, and that has to do with signals from the microenvironment. Whether it's environmental stresses, whether it's stromal cells, whether it's factors, growth factors that are in the area. There are attractor states that allow the cells to sit either in one state or another for different periods of time. And so this is part of what contributes to this type of a distribution that I'm showing you. Okay. So, I've given you an example of stochastic variation that has to do with transcription. But that's not the only level within a cell that can generate stochastic variation. As I've written up here, translation, post-translation, protein degradation, protein stability, epigenetic modifications, anything that modulates that amount of protein can undergo stochastic variation. Many times, these different processes balance out. But sometimes you can have a perfect storm, and you can have several of them go in the same direction, and you can have a big spike of protein. This has been measured. It's been seen in bacteria and yeast and mammalian cells. And then, for instance, take the example where I was talking about the replication proteins. Now you've got a cell that's just loaded with these replication proteins. It's going to have a very high probability of undergoing a duplication event or an amplification. So this is simply showing you what I was telling you before, that this is a very dynamic process and that different cells have different frequencies at which they find their set points, and that trying to understand the processes that govern this are very important, not only for phenotypes, but also for genotypes. And if one looks at amplification, it's been known for a long time that you can treat with these agents which interrupt replication. You can treat with environmental agents or pesticides, etc., and you can increase the frequency of these events by orders of magnitude. And so what I've tried to leave you with is this dynamism, this dynamic process that's occurring. And if you have this in mind, it's very easy to imagine how these processes can create the phenotypic variability as well as the genotypic variability that one sees in early malignancy. So part one, we can measure the frequency of ongoing plasticity in viable cells. The frequency of genomic plasticity increases with malignant progression is what we're finding. It can be governed by genetic and environmental modulators, and we now have a system where we can actually study these types of things. I wanted to say that cells under stress activate these mechanisms to accelerate evolution. And this description that I'm providing here is for bacteria, but could just as easily describe what happens to human cells when they undergo exposure to stresses. There's an increase in the rate of genetic exchange, mutations, there's resistance to all types of situations, there's there's remodeling of the environment in which they find themselves, there's motility to move to other places, modulation of immune surveillance. And when this happens, what it does is it causes specialization in a homogeneous population. The population now becomes heterogeneous. It actually turns on genomic plasticity. The whole purpose is to make specialized cells or different cells. Because when you have different cells is when you can now act with your companions and start to set up interactions within the community that allow for emergent properties such as competition, cooperation, etc. So I'm going to skip this example and just go to one that we're all familiar with. This is a forest. We know that tall trees can actually act in two ways. It can be very spiteful in terms of other trees that want sunlight because it can shield the sunlight, and so they can't get it. So that's a competition. But we also know that these trees can act in a very altruistic way to some of the bushes that are down there that don't actually want sunlight. They want the shade. So one tree can act in many different ways, just as one person in a community can act in many different ways with your brothers and sisters, so to speak. And we think that these types of interactions are probably at play very early in the malignant process and in disease states. And that this is probably what genomic plasticity is aiming for, to generate this diversity. And so bacteria, yeast, mammalian cells are able to turn on this process and cause these changes to occur. Okay? So there's fluctuations, and there are phase transitions going back and forth. Very briefly, I wanted to tell you about downstream consequences of genomic plasticity. It's the gift that keeps on giving. And I can summarize this part quite easily. And the reason that I chose this picture is because this one event of genomic plasticity can actually ripple outwards in the community and can actually come back on itself. So for instance, here's a tissue now that has undergone damage. And what happens is the cell talks to other cells in the environment, and you can see this red blush here indicating that these cells have responded to this stress. Okay? We've actually looked at this. We've identified the molecules that allow the cells to talk to each other. So when you have a break that occurs in the DNA, we've identified the pathway, p53 dependent, produces this diffusible factor. The diffusible factor can now interact with neighboring cells. I'm going to show you 3 different types. And what it does in this neighboring cell is that it turns on this stress response, stress-activated kinase, p38, turns on cyclooxygenase 2, produces prostaglandins, which can also diffuse, and it has differential effects depending on the cell. So the example that I just showed you, when there's damage, this is released, and it causes a normal cell to arrest, which is what I'm showing here. But you can have the exact same thing happening, but if these diffusible factors are actually bathe a cell that has repressed P16, instead of getting an arrest, you get the exact opposite. Now you get a stimulation of proliferation. So instead of one brown cell here, you'll have many, many brown cells. And so here I'm showing this, the damage. It goes out into the environment. The majority of the cells arrest, but there are rare cells around that have this now sustained stress response. We've captured these cells in the human body. This is the molecule cyclooxygenase 2. We can find pockets within tissues, whether it's pancreas, breast, prostate, whatever, that have turned on these processes. And it turns out that premalignant lesions always come out of these little nests of cells. And we found histological evidence to indicate that that's the case as well. How about other cells in the breast? What I just told you is about 2 types of epithelial cells. But how about the fibroblasts in the stroma that are around there? It turns out that when this damage is turned on in an epithelial cell, it talks to its neighboring stromal cells who have not undergone any damage, right? And this is what it turns on. We've named this program SEEP. It's a stress-elicited extrinsic program. And what you can see is that it acts together in a coordinated fashion to create a pro-tumorigenic niche. Notice many things that people have been talking about. Enhanced motility, enhanced angiogenesis, promoting inflammation, altered metabolism, deposition, remodeling of the matrix. So you can have a damage that occurs in an epithelial cell, and through these secreted factors, it turns on this whole process within the fibroblasts. It doesn't stop there. One of the things that it does is it increases the motility of the fibroblasts. And not only are these fibroblasts producing these factors, cytokines, etc., they're also producing mutagens, ROS, and many other things. So in essence, what you have is a little mobile tank, if you will, spitting out mutagens and causing more genomic plasticity as it passes through that environment. And it keeps cascading. That's why I showed you the drop and the ripples in the pond, because one damage can ripple throughout the community. And what I want you to focus on is this feed-forward loop. So the damage can cause these types of changes. I just told you that a response can be tissue remodeling, and this stress-elicited exogenous program can then feed back. So it can keep going. And if you have exposure to DNA damage, these little ripples start to accumulate, and now you can start to have synergy where they start to overlap. And it turns out that this synergy is very important because one event or another event on its own doesn't reach a critical threshold. But if you have enough of them and they're close together, now you have the cells that have the opportunity to start working together to go forward to create a disease state. So this amplifies the secreted signal to the adjacent cells, which now exhibit cause or consequence, whether it accompanies. There's debate. There's people on both sides of the question. But it seems to me that whether it is a cause or a consequence, trying to understand which cells are in this state and which cells have exited this state would be very useful. And we found evidence that that's actually the case. So in the last few minutes, I just wanted to tell you how you can listen in on what the cells are saying to each other and actually use it as a prognostic assay. So how can information about how a cell responds to stress provide clinical insights? And what I want to do is to tell you about this story, which is a very important clinical question. We know that by the time you actually see cancer, it's been around for quite a while, and it would be very useful to try and catch it in its earliest state. And many people are looking at fully formed tumors and doing expression profiling, etc. What I'm going to show you is that you can actually look at the tissues and how they're talking to each other here, and it will tell you whether this tissue is going to make a tumor 10 years in the future or whether it's not. Okay, so this is a very important question because it turns out that many people are diagnosed with premalignancies, but only 1 out of 10, roughly, about 12%, will actually get a cancer. yet everybody is treated as if they have a cancer. So let's take ductal carcinoma in situ. It used to be a rare diagnosis, but now it's not, and that's because of the advent of mammography. Here in the early 80s, you can see mammography became popular, and you can see this vast increase in the diagnosis of DCIS, and this line just keeps going up because of aging and because of the increased sensitivity of our equipment. So many people are riddled with premalignancies, but not everybody gets cancer. And as I said before, only about 1 out of 10 people that are diagnosed with DCIS actually get invasive breast cancer. But if it was 30 years ago, if you were diagnosed with DCIS, it was recommended that you get a mastectomy. That meant that a lot of people were being overtreated. Here is one example of what I'm talking about. If a woman has a mass, she is suggested to have a biopsy, and either she's diagnosed with breast cancer or she's diagnosed with one of these two conditions. Benign breast disease means that it looks a little bit funny, but it doesn't have a high association with future risk for breast cancer. But if you're diagnosed with DCIS or atypia, it has about a 12% chance of going forward. So a woman that is diagnosed with DCIS and has that little lesion taken out, 70% of those women, nothing will happen to them in the future. About 12% of them will get invasive breast cancer, and the remainder of this yellow part of the pie will get a subsequent DCIS. So what's important is if you can identify that fraction of women that are going to go forward and get an invasive breast cancer, they would have a very aggressive therapy. The ones that nothing is going to happen, they shouldn't have to have a full mastectomy or radiation or hormones. You should be able to identify those people and save them this problem. Okay, so people have been trying to figure that out for several years, and what pathologists have done is they've looked at many aspects of premalignant lesions and find that none of these aspects stratify for risk. Epidemiologists have looked at this. They've looked at weight, alcohol, ethnicity, etc. None of these variables stratify for risk. Molecular biologists have looked, and they've shown that nothing stratifies for risk. So in order to answer this question, to identify those that are going to go forward and very cleanly identify those that will not go forward, you need a good cohort, and we're going to talk about that tomorrow in terms of clinical samples, why you need these types of things, and you need some interesting ideas about how to approach this. So we approached this in our SPORE at UCSF, collaborated with Carla Kierlikowski and two trainees in my laboratory. And Carla had been collecting biopsies for the past 20 years. And just as I said, about 30% of them, about half are in situ and half are invasive. These women were diagnosed with DCIS, treated by lumpectomy alone, when there were no hormones or radiation as standard of care. And she has outcome for 20 years. And she tried all of the things that I just mentioned to you, and her conclusion was that nothing stratified. Nothing would predict who was going to do what. So what we did is we took the biology of these markers, this biology of stress response that I've been describing to you, and we asked if it could provide any information. And what I want to remind you is that the cells of our body are exposed to damage all the time, and they have ways of dealing with this stress. They have barriers. Here I'm showing senescence, apoptosis, and arrest. And we know that these are important because when we lose one of these barriers, we have increased malignant transformation. So one could hypothesize that, that perhaps the activation of these barriers would indicate that a lesion would not go forward because it's put on the brakes, so to speak. And one could imagine that if it looks as if the stress response is altered in some way, that these would be the lesions that would go forward because they've lost their proper braking mechanisms. And so we wanted to test this hypothesis, and to do so we looked at the senescence barrier and used the overexpression of P16 as a marker. But before you do that, you have to realize that P16 is repressed by RB. So there's two ways that you can get an upregulation of P16. One is that you have an activation of a barrier. That would be high P16 with no proliferation. That would mean senescence. And in our hypothesis, that would mean that you would not get a future cancer. Or you could have high P16 with proliferation, and that would mean that what you had done is to mutate RB, and now you promiscuously enter the S phase of the cell cycle. And so this would mean that second category where you would bypass senescence. So we use these markers, P16 and proliferation, and ask if they would stratify risk in this set of biopsies that Carla had been collecting for 20 years. And here was some of the first data that we obtained. It was really quite dramatic. Here's low expression of P16, and here's high expression of P16. But this is what tells the story. If the biopsy had high P16 and no proliferation, meaning that it had activated this stress response, there was a very, very low probability that these people would go forward to have a future tumor event. If these women had biopsies that had high P16 and high Ki67, meaning they had an aberrant stress response, you can see that literally every single one of them went forward, and I can tell you that the vast majority of them went forward to an invasive breast cancer, not a simple in situ DCIS. And these markers by themselves don't stratify, and you wouldn't expect them to, because it's the context that's important for this. So if high P16 and proliferation indicates that you're going to get a future tumor, what kind of a future tumor are you going to get? And so what we did is we looked at 130 tumors and asked, where do you find this little expression? And the answer was actually quite dramatic. You find it specifically in the basal-like breast cancers. Here's the high P16, here's the high proliferation. You do not find that combination anyplace else in this 120 breast cancers. These are the most lethal, the most metastatic breast cancers that there are. And as I indicated previously, you would expect that if you have high P16 in the presence of proliferation, you would have a problem with RB, as shown here, and you would have promiscuous activation of downstream markers such as cyclin E1, E2F, and that's what you have here. So deregulation of the P16RB pathway, this aberrant stress response, actually helps you to identify premalignant lesions that are going to become full-fledged tumors up to 10 years down the road. We could harvest the material for other markers. Another one that came up is cyclooxygenase 2. I'm mentioning that because I just told you in the previous segment that COX-2 is one of the linchpins for turning on this whole community response to stress. And it turns out that COX-2 is very important in terms of turning on pro-tumorigenic phenotypes, and it also allows for a very nice stratification. So I'm telling you that an abrogated response to stress actually can tell you what's going to happen to a future of a premalignant lesion. And what we've been doing is starting to develop markers to tell us which lesions within the breast have not yet activated COX-2 and which ones have this aberrant stress response. We've put together these biological markers, and we have an assay that is now being prepared for clinical use by patients and clinicians that stratifies risk for subsequent tumors. Women who express these markers, there are about 12 markers, they have less than a 3% chance of getting a future breast cancer. The reason that that's so exciting is that any person in this room with no unusual susceptibility has about a 6% chance. So to know that you have a 3% chance, that's pretty good. And the biopsies that express these markers, very, very high probability of going forward to having an invasive cancer. So what I've told you is that by listening to what the cells are saying to each other and how they respond to stress, we can probably use that information in a clinically useful manner. Can we modulate this conversation? That's something that would be very interesting to find out. Using the cellular response to stress, we've developed this assay that stratifies risk, and this assay tells us about cell-cell interactions and genomic plasticity. So I've told you about these three areas, and I'd like to thank the people that were involved in this work that I've presented to you. Thank you.
Thank you, Thea. That was wonderful. Great way to kick off the meeting. Questions? Yes, please. And please identify yourself. Oh, yeah. Since we have the webcast going on, if you don't mind using the microphone, that would be very much appreciated.
Yeah, thank you. Terrific talk. Really enjoyed it. Two questions. One, how is SEEP similar to or different from SLAM of Susan Rosenberg with stressful lifestyle associated mutations? Is one cell population based while the other is more population of bacteria? Is it the same thing one's dealing with?
I've thought for many, many years that there's beautiful parallels between bacterial interactions and mammalian interactions. And so there's the well-known SOS response.
Yeah.
I've found every parallel to that in mammalian cells. I call it the Mayday response.
Okay.
So I think that there are many commonalities. A question that you might also ask is how does SEEP relate to SASP, which is the secretory senescence program that Judy Campisi has identified. Okay. And what we find is that the timing is different, many of the factors are different, the cells that are involved are different, and the resolution is different. So this is now a brand new pathway that is immediately triggered under conditions where you have breakage within the genome, and what it does is it remodels the neighboring cells.
Okay, so the next question really comes down to what sets the evolutionary rheostat?
That's beautiful. And that's a question that we're very interested in. I think the first way to approach that is to start to identify some of the genetic determinants which affect those arrows that I was mentioning before.
And do de novo mutation rates suggest, for instance, that the rheostat will be related to fluctuation in that, for instance, copy number at 10 to the minus 4th versus point mutations at 10 to the minus 8th?
So in the story that I've told you today, I believe in the power of and rather than one or the other. Yes. I think the cell has many mechanisms at its disposal to accomplish the ends that it wishes. And I think that this pathway is a very exciting one that was heretofore unappreciated, but I wouldn't say that many of the other ones have no importance.
Thank you, Thea. That was a great talk. And I had a question, and I'm not sure I understood the conversation that just took place and whether my question may have just been answered, but because that idea of a rheostat, that I understood. So you started off by describing the stochasticity and all these different stochastic ways in which some of this genomic plasticity might occur. And then you show this series of examples where there's this reversion back to the mean, which is that being driven purely by statistics and stochasticity, or is there, I mean, that was implied, and you're saying that there's variation in the rate, so that implies that there is some kind of rheostat, there is some kind of control mechanism on that, and I just was wondering how well understood that process by which there is this, the different cells do have some kind of set point that they try to go back to.
So I think that's really interesting, and the way I find very useful for thinking about this, please don't laugh too hard, is I think of standing on a trampoline. And so it's kind of mushy, and if somebody has taken a part of the trampoline and pulled it down quite dramatically, there's a really good chance that while you're bouncing around, you're going to end up in that hole, right? So there can be peaks which you wouldn't necessarily land on, and there can be valleys where it would be very easy to fall into. And all of this is dictated by the environment, by the agents that are there, by the stromal cells, the cytokines that are there, by mutations that exist within the genome. That creates what the trampoline looks like. And then what the cell does is kind of bounces around. If it's a cell that has undergone differentiation and is a wild-type cell, its bounce is pretty slow and, it doesn't have a purpose. What we found is that if you treat with DNA damaging agents, environmental stressors, it's almost like turning up the heat. Now it starts bouncing around quite a lot, and it starts exploring other states in a very active manner.
But it's that interaction between that disposition in the cell and the stromal environment of the cell culture or whatever system you have. Okay.
Yes.
Thank you.
Hi, Helmut Zarbel. I'd like to get back to the issue of the stem cells. So if you're talking about plasticity both in phenotype and genotype, you can, and you referenced NAGOG, that seemed to be controlling whether the cells are can differentiate or not. What about repair enzymes? So if you have a lot of plasticity in the expression of repair enzymes in a stem cell, mutations in stem cells are probably of greater consequence than differentiated cells. So is that under tighter control in the stem cells? Is there any evidence that you have less heterogeneity?
So that's a beautiful question. And I made an allusion to that when I said that different loci have different rates. So there are some areas where you would want it under tight control and other areas where actually you really want it to flail around and test things. When you start to think about the things that we're talking about today, all three of these actually have something in common. They're reversible. A retrotransposon can go into a site, but it can come out of the site. Gene amplification, otherwise known as copy number variation, can go up and down, quite actively. That's been shown for many, many years. And telomere length, I think we're going to hear some beautiful talks showing how what we once thought was, the telomeres getting shorter and shorter and shorter. That's not the case. They can zip all the way up to very, very long. There are environmental or emotional conditions that can make them get very short, and then they can reverse themselves. So it's a very dynamic part of the genome that can react to the environment in which it finds itself.
Hi, Dave Gilley. You started talking about the resetting the rheostat, or once you selected a population at some spectrum, it came back to reset, and that the importance of looking at individual cells. But in your very exciting results from the DCIS types that go on to malignancy, it seems like that rheostat is kind of off in some way, and that the population's not resetting back to sort of a homeostatic level of COX-2 and P16.
So when I describe the homeostatic heterogeneity, the reacquisition of that spectrum, those cells were in a given condition. What we know is that the stromal conditions and the community conditions within a premalignant lesion are anything but static and homogeneous. And so I think what we're looking at is a beautiful manifestation of the interaction between the two.
Mm-hmm.
I just had one last question. First of all, thank you again. That was wonderful. Could you put this in the context for us of the mutator hypothesis as an evolutionary driver of cancer? Would it be that the quote-unquote mutator genes that we're looking for might in fact be those involved in the rheostat, or keeping cells in a compartment where they are more or less likely now to accumulate the genetic alterations that push them along the process?
I think Larry Loeb's discussion of mutator phenotype was beautiful. It's very scholarly, and it really provokes thinking about how these things might happen. And as I understand the mutator phenotype, it's the misregulation of repair enzymes, et cetera, that would allow the accumulation.
As it was understood.
Right, as it was understood. I'm trying to be respectful. And you get the accumulation of mutations, which can now explain what many thought was a multi-hit hypothesis. What I've tried to describe here is actually fundamentally very, very different. What I've tried to describe is a dynamic process whereby cells within a community can explore different possibilities. If it's not a good possibility, that cell dies, you still have the community. It still has plenty to do with what it wants. If it turns out that that's very helpful, it can enjoy a proliferative advantage. But the things that I'm talking about are transient, and they can go back and forth. That's why I said retrotransposons, telomeres, and copy number variations are beautiful examples that all fall into this category. That's different than what one might think of for, say, point mutations. I don't put deletions in the same case as point mutations because actually there's evidence that you can rescue a deletion with, the other allele. So there is the potential for reversibility there. So I think that this is fundamentally different than what Larry Loeb has been talking about, and I see it put to a different use as well. I think that this is important in evolution, differentiation, and overall response to what's happening in the environment.
Thank you very much. All right. That was a great kickoff for session 1.